Here's a question that we've asked with each of these three different phases of cellular respiration. With glycolysis, you could name the forms of energy that enters glycolysis. Same thing with the Krebs cycle. So we're doing this for the electron transport system. How does energy enter into the electron transport system? So there's no sugar at this point. All it is hydrogens delivered by these two hydrogen carriers. What form does energy leave the electron transport system? So at this point, there's no more intermediates. There's no pyruvate or acetyl-CoA or hydrogens or anything like that. There's no electrons. This has all resulted in the synthesis of ATP. That's the final energy product of the electron transport system, and there's a lot of it. There's 32 ATP that are made, which is quite a few. So one molecule of glucose in most cells can be used to make 36 ATP, 2 in glycolysis, 2 in the Krebs cycle, and 32. That's a lot more than in glycolysis in the Krebs cycle. There's some cells that can only make 32. There's some cells that can make 38 ATP. It just depends on the organism. But the typical number is 32 ATP that can be made in the electron transport system. Okay, so another thing that I've done each step of the way, these three different phases of cellular respiration that I've tried to do to make this easier on you, because there's a lot of information, there's a lot of terms, a lot of figures with labels, is to point out what's important. So in the electron transport system, these first two, the hydrogen carriers, NADH and FADH2, those are how hydrogens are delivered to the electron transport system in the first place. It's what starts the whole thing. Then hydrogens are split into their atomic components, electrons. Electron adds energy that's used to create a proton gradient to make ATP. Another thing that we know is that we breathe in oxygen. This is where oxygen is used. That's number 7. And that oxygen, we can't take in an endless supply of oxygen either. We've got to get rid of that oxygen. We don't breathe it out as carbon dioxide. We take those protons, electrons, hydrogen, combine it with oxygen, and make water. And we're constantly losing water. We make a certain amount of metabolic water. We have to drink a lot of water. There's some animals that live out in the desert that make water metabolically like this. They carry out cellular respiration in the electron transport system in order to make their own water. And they burn the fat that's got a lot of calories, a lot of production of water, because they live in the desert and water is a shortage. So that's what's happening there. So in terms of cellular respiration in general, if you could draw this figure one way or the other, if you could take glucose, just write down glucose and then write down these other things, pyruvic acid, acetyl-CoA, if you're especially having these arrows go out, NADH, ATP, acetyl-CoA, carbon dioxide, if you can follow these things. So a figure like this that doesn't have very many parts to it, glucose, pyruvic, acetyl-CoA, carbon dioxide, ATP, NADH, FADH2, ATP. And if you added to this, understanding what happens in the electron transport system, that you've got hydrogens that within this electron transport system rectangle, then you'd have electrons and you'd have proton in the form of a proton gradient representing energy. And then if you added this, the fate of oxygen picking up the hydrogens, basically, electrons first, electron receptor, then that's pretty much summarizes cellular respiration. We've spent a lot of time talking about it, 3 days, talking about the cellular respiration. But you can boil it down to some pretty pretty limited number of terms that are really important, especially concentrated on energy, one, and these other things that we're very familiar with, like carbon dioxide and oxygen and water. So you could also, we've seen this table before, this figure. You could also construct this, say a glycolysis. Okay, how does energy come in? Is glucose. How does it go out? ATP, NADH, pyruvate. Pyruvate goes in, or you could change pyruvate to acetyl-CoA and fill in these boxes, each one of these. You could construct something and test yourself by having to fill these boxes in and having to say, how does any energy enter in and how does energy exit each one of these? Something like this. All right, well, one final thing, and then we'll be done with this whole unit, is that we don't just eat glucose, obviously. We eat bread and we eat pasta and we eat all kinds of donuts and cereal and we eat chicken and we eat pork and we eat beef. We eat all kinds of different things. What we've looked at is something comes out of starch, or that we break down from glycogen that's stored in our muscles. We've looked at this, glucose going into glycolysis, the Krebs cycle, the electron transport system, making a bunch of ATP with the end products, carbon dioxide and water, those waste products. With lipids, it's a different, we'll go all the way over here to lipids. Lipids aren't glucose, obviously, but lipids have two different parts. They've got this glycerol molecule, which can enter into glycolysis, and they've got these fatty acids, these carbon chains. They've got 20 carbons, or 22 carbons, 18 carbons, 16 carbons. Different fatty acids have different numbers of carbons. So in our digestive systems, we can break these fatty acids down into two carbon chains, which is very similar to acetyl-CoA. So the fatty acids don't go through glycolysis. The fatty acids skip that, and then as modified into acetyl-CoA, 2 carbons at a time, they enter into the Krebs cycle. Then the hydrogens go to the electron transport system. And the waste products of lipids, again, are water and carbon dioxide. Proteins is a different situation where there's no glycolysis that's going on. These amino acids, again, are chains of carbon. There's a bunch of carbons that are joined together. We've got enzymes that can break those proteins down, those amino acids, break them down into 2 carbon chains. Those 2 carbon chains enter into the Krebs cycle as acetyl-CoA, and then to the electron transport system, and making a whole bunch of ATP and producing carbon dioxide and water. But the other thing, you guys all know that every single protein has nitrogen in it. There's not nitrogen in carbohydrates. There's not nitrogen in lipids. So these nitrogens build up when protein is broken down. The carbons and the oxygens and the hydrogens in protein, they go the same pathway as they do in carbohydrates and lipids, with the exception of not going through glycolysis. But they end up as water and carbon dioxide. These proteins that contain lipid or nitrogen, then nitrogens have to be dealt with. And so humans, for example, we'll just use that as an example since we're all human. We store nitrogenous wastes as urea, and then we eliminate urea in our urinary system or our excretory system. We've got to use some water to get rid of all that nitrogenous waste. We produced all that nitrogenous waste because we break down proteins. We eat protein. We produce a lot of nitrogenous wastes. You need some excretory system or some mechanism for getting rid of all that nitrogenous waste, and that's exactly what happens. So carbohydrates, clean burning, in the sense that carbon dioxide and water is produced. Lipids, clean burning in the sense that carbon dioxide and water is produced. Proteins, not clean burning because they produce this waste, nitrogenous waste. All right, so we've just looked at very simple glucose as our fuel, but it is similar for these other types of sources of energy besides simple carbohydrate, simple sugar like glucose. And they can enter into these same metabolic pathways and they can be used to make ATP. So that concludes this electron transport system lecture. If you go back through your notes, go back, look in the syllabus, look in the study guides, then it should be obvious what we finished on for exam 1 and where the material for exam 2 starts. That's what's going to be on the exam on Wednesday. There's a lot of information here in the lectures, a lot of information in the study guides, and you can test yourself in the practice questions after you've studied to see how well prepared you are. Then everybody should come in. You guys did a very good job of this last time. This exam is going to be harder, but you should come in and have a very good idea what kind of things I'm going to ask you about, because it's going to be just what I lectured about. It's going to be on topics that are included in the study guides, and they're going to be questions that are similar, sometimes very similar, but maybe a lot of them not exactly the same, certainly, as the questions in the practice questions. And then you've been doing mastering biology along the way, so you should have a pretty good hand on this, pretty good handle. Okay, so I'll see you guys on Wednesday.